Welcome to our video lecture on cellular respiration. Our essential understanding for today is that respiration supplies energy for the functions of life. Our objectives: we are going to understand the importance of ATP to those functions of life. We're going to describe the structure of mitochondria and how it relates to its function, making ATP. We're going to describe glycolysis, we're going to outline anaerobic respiration, and then we're going to look at the processes of aerobic respiration, which are the link reaction, Krebs cycle, and oxidative phosphorylation, sometimes also referred to as the electron transport chain. We're going to wrap up with a quick explanation of how respirometers can be used to investigate the rate of respiration. So imagine, if you will, I've got a marshmallow right here, lots of sugar in my marshmallow, and I hold it over a campfire. Oxygen from the atmosphere, energy from the campfire, causes this guy to set on fire, and the sugar molecules in that marshmallow release lots and lots of energy, and we end up with some carbon dioxide and water. We basically do the same thing in our mitochondria, except instead of having big fire, big energy released all at once, we have these tiny little steps of energy being released, and then we can store that energy in the form of ATP. We're still going to start with our sugar and our oxygen. It is still kind of sort of a combustion reaction. We still form carbon dioxide and water at the end. It's just a matter of tiny little steps in our mitochondria, or all at once in our campfire. So what even is ATP? ATP is our energy source for all the functions of life, that cellular activity that we carry out. We have this nitrogenous base that's known as, if I could spell, adenine. We've got our ribose sugar, and you're probably thinking, hey, isn't this a nucleotide? Yes, my friends, yes it is. And it has 1, 2, 3 phosphate groups, and that's what makes it ATP. T for triphosphate, 1, 2, 3 phosphate groups. What we do with that ATP is we actually chop off one of those phosphate groups. We chop off that phosphate group. I'm going to not draw over its title. And we call that phosphate group by itself inorganic phosphate, or PI. So PI is this phosphate group by itself. When I chop that phosphate group off, the energy that was stored in that bond between the phosphate groups is actually transferred to enzymes, or whatever other protein needs that energy. And then my ATP now turns into ADP because that phosphate group got transferred to whatever. So those 2 phosphate groups remain. That's ADP. Interestingly, we could chop off one more phosphate group. I could be left with just one, and that would be AMP, adenine monophosphate. This phosphate group can actually get put back onto ADP and turned back into ATP, and that's exactly what we do in cellular respiration. So we're going to take a look at the mitochondria, the structure and function of the mitochondria. Mitochondria have 2 membranes, and we think that outer membrane, this outer membrane, was formed during endocytosis, that endosymbiotic theory. So the outer membrane contains transport proteins for getting stuff into the mitochondria, most importantly pyruvate, and we'll talk about what pyruvate is in a couple more slides. The inner membrane of the mitochondria contains the electron transport chain and ATP synthase. These are all proteins, protein complexes. They are embedded, like literally stuck inside that inner membrane. And what's super cool about that inner membrane is that it has these cristae, or folds. So all of these folds of the inner membrane just increase the surface area. So we have lots more space for all of those proteins of the electron transport chain and for ATP synthase. The intermembrane space is literally the space between the membranes, intermembrane space. What's super cool is that that space is pretty small. We're going to pump some protons or hydrogen ions into that space. The fact that that space is small means that it fills up pretty fast. So this is good because we can make the proton concentration gradient that we need super quickly because the space is small. On the inside of the inner membrane's inside space, this is known as the matrix. The matrix has all the enzymes needed and the pH necessary for Krebs cycle. Krebs cycle occurs in the matrix. So these are all the pieces and parts of the mitochondria. So go ahead and take a moment, find those pieces in the diagram, and then also find those pieces in this micrograph, this electron micrograph. Good. All right, so hopefully you remember that the matrix is here in the middle. These folds are known as cristae, and they are the folds of the inner membrane. So that inner membrane goes all the way around, and it's highly folded into all those cristae, so that we have lots of surface area for all of our proteins of the electron transport chain and ATP synthase. We also have that outer membrane. It has proteins needed to transport pyruvate into the matrix of the mitochondria. And then we also have this super cool space between the two membranes known as the intermembrane space. And it's nice and small so that it's easy for us to accumulate or build up a high concentration of protons or hydrogen ions. On the electron micrograph, so we definitely can see an outer membrane. I'm going to switch up colors here. All of these folds are folds of the inner membrane. So these are all the cristae of the inner membrane. Here we have the matrix, the middle of the inner membrane, and then these tiny little spaces in between, in the middle of the cristae, that is our intermembrane space, the space between the membranes. Good. All right. So cellular respiration, crazy big overview, lots of words. This is going to be super confusing until we go through this a couple times. But quick look at what's going to happen, what we're going to talk about in the next couple slides. So cellular respiration, an overview. We're going to start with this guy, glucose. We're going to break glucose down in the process of glycolysis into this molecule, which is known as pyruvate. If we don't have enough oxygen available, we're going to move into anaerobic respiration. Anaerobic respiration is also known as fermentation. And if you are us, humans, mammals, you're going to turn that pyruvate into lactic acid and a little bit of NAD+, which can be used in the next round of glycolysis. If you're super cool like a yeast, then in fermentation you can make some ethanol and some carbon dioxide and also some NAD+, which again can be used in glycolysis. What we love about glycolysis is that we do make 2 ATP. That is super important because we get a little bit of energy out of that first step of breaking up the glucose. If we have sufficient oxygen available, instead of going to anaerobic respiration, we're going to do instead aerobic respiration. That pyruvate that we made during glycolysis is going to now be transferred into the mitochondria. Everything that we already talked about here happened in the cytoplasm of the cell. We're going to move that pyruvate into the mitochondria now, into the matrix of the mitochondria. The first thing that happens is the link reaction. The link reaction is going to chop off one of those CO2s from this molecule. What's left is a thing called an acetyl group. We're going to add that acetyl group to a coenzyme called CoA. We're going to form acetyl-CoA. Acetyl-CoA enters Krebs cycle. We're going to chop off some more carbon dioxides. We're going to make some more NADH molecules, a little bit of FADH2 and a little bit of ATP. The important thing that happens during Krebs cycle is the formation of all this NADH and FADH2. These guys are known as electron carrier molecules, and I really don't like that name because they carry electrons and protons. So electrons and protons on these molecules. They're going to carry them to the electron transport chain. The electron transport chain literally is going to transport electrons. So we're going to take those electrons that were carried by NADH, and we're going to drop them off on these proteins that are embedded in the inner membrane of the mitochondria, the proteins of the electron transport chain. The electrons are going to jump, jump, jump along these proteins. Energy from the electrons is going to be used to pump hydrogen ions from the matrix into the intermembrane space. So we get this super high concentration of hydrogen ions in the intermembrane space. The universe does not like for things to be hoarded up like this, and so those hydrogen ions are going to look for a path back into the matrix. Because they have a charge, they cannot go through the phospholipid bilayer, and so their only path is through these proteins called ATP synthase. ATP synthase. And when those hydrogen ions flow through ATP synthase, this protein can take ADP, adenosine diphosphate, and an inorganic phosphate group, glue them together to make some ATP. For every one glucose molecule that we start with, 34 ATP can be made doing this process of electron transport chain. We call it oxidative phosphorylation because we're doing some oxidation, which is losing electrons, and phosphorylation, which just means adding a phosphate group. That's a lot, I know. Luckily, I'm going to repeat all of it in the next 10 slides. All right, so back to glycolysis, that first step of respiration. So glycolysis is just taking glucose, and we show it with these 6 balls because glucose is a 6-carbon sugar. So we've got 6 carbons here. We're going to go through glycolysis, which literally means splitting up the sugars. We're going to end up with 2 pieces that are each only 3 carbons big, and that's called pyruvate. We're going to use 2 ATP to make this happen. So we're going to add a phosphate group here, add a phosphate group here. This adds a bunch of energy, which causes this guy to split in half. We're going to take 2 NAD+ molecules. We're going to add some electrons and some hydrogen ions that we pull from the middle of this glucose molecule, and we're going to make 2 NADHs. The H is because we added the H's. The plus sign goes away because when we add an electron, those guys go neutral. Positive, negative goes to neutral. We're interestingly also going to make 4 ATP. So we use 2 ATP and we get to make 4 ATP. So we have what we call a net gain of 2 ATP. So we already have more ATP than we started with, and we only did glycolysis. This happens in the cytoplasm of the cell. So let's say that we do not have a whole lot of oxygen available. We're going to take that pyruvate and we're going to do anaerobic respiration. Anaerobic respiration, also known as fermentation, we don't have oxygen. So anaero, no oxygen available, or not enough oxygen available. So in glycolysis, we used NAD+ to make NADH. And if I had oxygen, this would be super helpful. But if I don't have oxygen available, I want to turn my NADH back into NAD+ so that I can do glycolysis again, so I can make some ATP again. So this is why we go through anaerobic respiration, is to turn that NADH back into NAD+ so that I can do some more glycolysis. We have 2 options for this. If you're not cool, like mammals, then you take your pyruvate and you turn it into lactate or lactic acid. And this is what makes our muscles sore after we work out a little bit too hard. If you're super cool like a yeast, then you can take that pyruvate and you can chop off a CO2 molecule, and what's left over is ethanol. So again, we are going to start with glucose, 6 carbons, glycolysis, we're going to make pyruvate, which is 3 carbons big. Yeast will turn those 3 carbon pyruvate molecules into carbon dioxide and ethanol, and then we're going to turn that NADH back into NAD+ so that we can do some more glycolysis and make some more ATP. If you're not cool, you're a mammal, then you're going to take that pyruvate that was formed during glycolysis, you're going to turn it into lactate. But the good thing is you still turn some of that NADH back into NAD+ so that you can do glycolysis again, so you can make just a little bit of ATP, because a little bit of ATP is better than 0 ATP. So super cool yeast guys are going to make ethanol and CO2, and we humans have definitely taken advantage of that. So carbon dioxide bubbles, this is what causes our bread to rise. So the reason this guy is nice and fluffy, all the nooks and crannies in our bread come from the carbon dioxide that's formed when yeast in our bread dough is producing that CO2. Ethanol is also in our bread dough, but it evaporates when we bake the bread. So yeah, we bake the bread. We don't bake beverages though, and so we certainly can also take advantage of this process to make beverages that you can drink when you are 21. So back to glycolysis. Remember that in glycolysis, we're going to take that 6-carbon glucose, we're going to split it up, we're going to make pyruvate. We're going to make this little bit of NADH, we're going to make a little bit of ATP. Let's say this time around, now we do have oodles of oxygen gas available. We're going to move into aerobic respiration. Glycolysis occurs in the cytoplasm, so all of this is happening in the cytoplasm. Now, finally, we're going to start talking about what happens in the mitochondria. So the first thing that's going to happen, we're going to take that 3-carbon pyruvate molecule that's in the cytoplasm, which is also called cytosol. We're going to move that pyruvate into the matrix of the mitochondria. We're going to chop off a CO2 group. This is called decarboxylation. Decarboxylation, because I'm chopping off, that's the D part. Carboxyl, that's that CO2 part. So decarboxylation, we're chopping off CO2. What's kind of cool about every time we chop off a CO2, every time we break one of these molecules, we basically have little crumbs of hydrogens and electrons. So electrons and hydrogen ions are produced when I decarboxylate that pyruvate. Those electrons and hydrogen ions are grabbed by this molecule, NAD+, and that guy is turned into NADH. And we can say that it's also plus H+. We can call this the reduced form because it has gained those electrons and hydrogen ions. So we're going to take that pyruvate, decarboxylate it, take those electrons and hydrogen ions, chunk them onto NAD+, which forms NADH. Then what's left over is called an acetyl group. We're going to join that acetyl group to this coenzyme called CoA, and that's going to make this thing called acetyl-CoA. Acetyl has 2 carbons left. CoA has 4 carbons in CoA. Good? All right, so this is all happening in the matrix of the mitochondria. I now have this thing called acetyl-CoA that was formed from that pyruvate. I'm going to take that acetyl-CoA and we're going to send it through this thing called Krebs cycle, which is also called the tricarboxylic acid cycle, which is also called the citric acid cycle. We talk about Krebs most of the time, so Krebs will be what I use most of the time. So that acetyl-CoA that we made in the link reaction, that acetyl group is left over from the pyruvate. Remember that pyruvate came from glucose. So glycolysis turned glucose into pyruvate. The link reaction turned pyruvate into acetyl, and then we chunked that CoA molecule onto it. We then now enter citric acid cycle, also known as Krebs cycle. We're going to do a bunch of stuff, and you don't need to remember any of those names. Do not even panic about all those letters. What we need to know is that we're going to do some more decarboxylation. There's some more decarboxylation because we're chopping off those CO2s. We're also going to make some more NADH. And now we're going to add this other molecule, FADH2. The NADH, the FADH2, this is all these are all electron carrier molecules. And again, I don't like that name because they also carry protons, proton and electron carrier molecules. This is the important stuff that's getting formed. So this NADH and this NADH and FADH2 that we're making every time we decarboxylate that stuff that's left over from the glucose molecule that we started with a long time ago, this NADH, FADH2 is going to carry, that's why we call them carrier molecules, carry those hydrogen ions and electrons to the electron transport chain. So all of this buildup has gotten us to this point where we're looking at the electron transport chain. Here is our outer membrane of the mitochondria. Here's our inner membrane of the mitochondria. Between those membranes is our intermembrane space. Here we're showing that it's folded, so there's the cristae. This is the matrix of the mitochondria. What's going to happen is this, that NADH and FADH2. These guys that were formed in Krebs cycle here in the matrix of the mitochondria, they're going to carry their electrons and protons. They're going to carry their electrons and protons to the proteins of the electron transport chain. the electrons are going to jump. The electrons are going to jump, 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 jump along the electron transport chain. Every time those electrons jump, a little bit of their energy is transferred to these proteins. These proteins use that energy to pump hydrogen ions from the matrix into the intermembrane space. So again, we have electrons on the NADH and the FADH2. Those electrons are going to jump, jump, jump along the electron transport chain. That energy from the electrons jumping is transferred to these proteins of the electron transport chain. The proteins of the electron transport chain are going to use that energy from the electrons jumping to take hydrogen ions and pump the hydrogen ions into the intermembrane space. So now I've got all of these hydrogen ions stuck in the intermembrane space. I have a high concentration gradient of hydrogen ions in the intermembrane space. The universe does not like for things to be all mashed up like that. So these hydrogen ions want to spread out. They want to go back to the matrix to get back to equilibrium. Their only path to get back, the only way that they can possibly get back into the matrix is through this protein called ATP synthase. So these hydrogen ions, all of these hydrogen ions that we just pumped into the intermembrane space, they are going to flow through ATP synthase. ATP synthase is going to use the movement energy, that kinetic energy of those hydrogen ions moving through it, to glue together some ADP and some inorganic phosphate. ADP, inorganic phosphate. And when these two guys get smashed together, we now have ATP. That ATP is used for everything that we need to do. Sounds good? Oxidative phosphorylation is what this whole thing is called. It's because we are phosphorylating. That just means I'm adding a phosphate group. Phosphorylation just means add a phosphate group, that inorganic phosphate. Oxidative comes from chemistry. So remember that in chemistry we have reduction, oxidation reactions. So oxidation is the loss of electrons. Because NADH and FADH2 are losing electrons to the electron transport chain, this is oxidation. So we call it oxidative, losing electrons, phosphorylation, add a phosphate group to the ADP to make the ATP. We also have this vocabulary word chemiosmosis. Remember that osmosis was that movement of water across a membrane? So chemiosmosis is the movement of a chemical across a membrane. And that's what's happening here. I have this high concentration gradient of hydrogen ions in the intermembrane space. They're going to move across the membrane. That's the osmosis part, because they're not water. It's chemiosmosis. They're going to move across that membrane from high concentration to low concentration in the matrix. That's chemiosmosis. This happens because we make this proton gradient. Proton gradient simply means that protons are the same things as H plus as hydrogen ions. So we're going to have a higher concentration of protons, hydrogen ions, in the intermembrane space. And so I have a gradient, high and low, of those proton concentrations because of the electron transport chain pumping all those hydrogen ions into the intermembrane space. One more little thing to look at on this slide, and then we're going to tell this story all over again. Oxygen is important. Remember, this is all about aerobic respiration. We need that oxygen for all of this to happen, and the reason is because those electrons that are jumping are electrons that are jumping. Electrons jump, jump, jump along the electron transport chain. When they get to the end, they have to go someplace so that the next electron can jump, jump, jump. If these electrons get stuck, there's no space for them to jump, and then the whole thing shuts down. So we have to take those electrons off of the electron transport chain, and that's the job of oxygen. So oxygen is going to grab those electrons at the end of the electron transport chain. It's also going to grab some hydrogen ions from the matrix, and together oxygen and hydrogen and electrons make water. We call oxygen the final electron acceptor because it literally accepts the electrons at the end, the final part of the electron transport chain. All right, I'm going to tell you that story all over again one more time because it's a lot. All right, so again, electron transport chain is embedded in the inner membrane of the mitochondria. So here we have our outer membrane, here we have our inner membrane, here you see those proteins of the electron transport chain all embedded in the inner membrane of the mitochondria. Here we have the matrix, here we have that intermembrane space, the space between the two membranes, right? Good. So what's going to happen? NADH is going to come along and drop off its electrons. FADH2 is also going to drop off those electrons. Where did NADH and FADH2 get their electrons? In Krebs cycle, and also a little bit in link. So we're going to take these electrons and they are going to jump along the electron transport chain. As those electrons are jumping along the electron transport chain, they are going to transfer their energy to these proteins of the electron transport chain. That energy that's now gained by these proteins is going to be used to take hydrogen ions also carried by NADH and FADH2. We're going to take these hydrogen ions and we're going to pump them into the intermembrane space. We're going to pump hydrogen ions into the intermembrane space. We're going to pump hydrogen ions into the intermembrane space. So now I've got all of these hydrogen ions all smashed up in the intermembrane space. This is our proton gradient. Proton gradient. The universe is not stable when you have a high concentration. We have a high concentration here and a low concentration in the matrix of the mitochondria. This is not stable, and so these hydrogen ions are going to flow back into the matrix to get to equilibrium. Their only path is through this protein complex called ATP synthase. ATP synthase is going to use the energy of those hydrogen ions moving to glue together ADP and inorganic phosphate to make ATP. This process is known as chemiosmosis. We have the osmosis of those hydrogen ions flowing through. We're going to couple the energy of that movement with this chemical reaction, ADP plus PI, to make ATP, chemiosmosis. Remember that at the very end of the electron transport chain, those electrons have to jump someplace else so that there's space for the next electron to jump. We're going to take those electrons, we're going to take some oxygen gas and some of the hydrogen ions that are also floating around in here, combine them all together to make water. This is oxidative phosphorylation. Oxidative because NADH lost electrons and then the electron transport chain lost electrons to the oxygen. We call it phosphorylation, because we're phosphorylating or adding a phosphate group to ADP to make that ATP. Amazing, right? All right, so how do we measure the rate of respiration? We can use this thing called a respirometer, which may or may not be ethical. So in this investigation, we've added some living organism. In here we have a cricket. The cricket is going to breathe in oxygen to carry out respiration to make some ATP. We're going to take this vial, add a tube. We're going to submerge this tube under the water. So now there's a little bit of water here. As the cricket uses the oxygen, the water is going to get pulled in, and then we can measure how fast the water is moving through the tube. That'll tell us how quickly the cricket is using that oxygen gas. Now you might be thinking, but wait, the cricket's also making CO2. So every O2 that comes in, a CO2 is going to go out, and won't that push the water back the other way? Yes, it would, except we're going to add some potassium hydroxide, also known as potash, and that CO2 is going to combine with this to make potassium hydrogen carbonate, which is a solid, which is just going to stick to whatever this material is. We usually throw a cotton ball in there. So we're actually going to get rid of that CO2 by combining it with this potash with potassium hydroxide. So the only change in gas volume is the consumption of that O2. Cool beans, right? unless you're the cricket stuck in this thing about to drown in the water. Hopefully the investigator is watching, and when the water gets to the cricket, we rescue the cricket and let him go. I hope that's what happens. All right, so we definitely have achieved our objectives. We talked about ATP, the importance of how it can transfer energy through that phosphate group to enzymes. Talked about the mitochondria. We described that first stage of all respiration, glycolysis. We talked about anaerobic respiration if we have a lack of oxygen gas. If we have lots of O2, then we go into aerobic respiration, which is the link reaction, and Krebs, and oxidative phosphorylation. And then we finally wrapped up with that very short talk about respirometers. Good work today, my friends.